USDA logo. United States Department of Agriculture. Meet the expert, George Paoli, PhD.
My name is George Paoli. I'm a research microbiologist and lead scientist at the USDA Agricultural Research Service, Eastern Regional Research Center in Windmore, Pennsylvania. I serve as a lead scientist on a project with two other scientists, Drs. Chin-Yi Chen and Galen Ulick.
Describe the subject area of your research.
We study Shiga toxin-producing E. coli, pathogenic E. coli, in biofilms and in microbial communities with interest in determining how the pathogenic E. coli persist, particularly in beef and beef processing facilities.
Why are you interested in studying Shiga toxin-producing E. coli?
We are interested in studying Shiga toxin-producing E. coli in beef and beef processing environments. We're interested in how the organism persists in those environments and makes its way into foods that we consume. Our goal is to learn how that organism persists and ultimately learn to control that organism and eliminate it or reduce its incidence in those environments.
Describe the use of any specialized equipment used in your laboratory.
One of the things we do use that is particularly special is with the advent of next generation DNA sequencing, we've been adopting that technology to look at microbial communities, and we work with this instrument. It's called the MySeq. It's the Illumina MySeq instrument. It allows us to look at a lot of different organisms, a lot of different DNA sequences at the same time. This instrument will provide 25 million sequence reads in a single run. If you're doing a bacterial genome, that might allow you to sequence about 10 genomes. But we can look at hundreds of microbial communities in a single analysis. So that allows us then to look at the communities of organisms that are present in, again, beef and beef processing facilities and try to attribute, maybe find a correlation of the organisms that are persisting in those environments with the presence of pathogenic organisms such as E. coli 015787.
What is most interesting about this work?
Well, one of the interesting things about E. coli, about bacteria in general, is that it's generally a commensal organism in the mammalian gut. So it really doesn't cause, most E. coli don't actually cause harm, they actually benefit the host. But certain of these organisms actually develop the ability to become pathogens. And what's particularly interesting about E. coli is that E. coli, commensal E. coli, is a rather good biofilm former. It can actually form attachments and communities on surfaces. But E. coli 015787, which is Shiga toxigenic producing, produces this toxin that it gives the organism its name. But the toxin is particularly important for pathogenesis. The gene for this toxin is actually mobile, and it's present on a bacteriophage or a virus that infects the organism. And in doing so, it integrates into a site that actually disrupts biofilm formation. So the question became, how is it, and when you look at these organisms, you find that the pathogenic forms are not particularly good biofilm formers. How are they persisting in these environments? So one of the things that we've done is, Dr. Ulick in particular, has studied a lot of taken the approach of really studying the physiology and the genetics of these organisms to better understand how they form biofilm and how they behave during biofilm formation. And he's discovered, we as a group, and he in particular, have discovered a number of genetic mechanisms that the organism has utilized to overcome this disrupted gene.
Are there any important findings from your research?
One of the approaches we are taking is to, instead of just looking at E. coli 015787, is to also look at the communities of organisms might be allowing that organism to persist in foods and in food processing environments. So we've sampled those environments and foods to look at those communities, and we've used the next generation sequencing metagenomic approach to identify those communities. We're now analyzing those communities both microbiologically to determine if we can find organisms that are allowing E. coli to persist, doing laboratory experiments to find those organisms. We're also looking at those communities to see if we can find indicator organisms that might allow us to determine whether E. coli is present based on the community rather than doing specific pathogen detection.
What are the next steps for your work?
One of the interesting things that we've learned in studying biofilm formation in chigotoxigenic E. coli is that the organism, as I mentioned, actually integrates a phage or into the genome to carry the toxin gene. Certain experiments were done to actually get that particular phage to pop back out of the genome, and in doing so, reconstituting biofilm formation. And what was very interesting in those studies is to find that not only was biofilm formation disrupted or restored to some degree, it had a dramatic effect on virulence gene expression. So we're very interested in following that up and looking at the relationship of biofilm and virulence gene expression and the potential for biofilm or cell adherence and attachment in both the bovine host and the human host, and looking at potential virulence.
USDA is an equal opportunity provider and employer. To file a complaint of discrimination, write to USDA, Assistant Secretary for Civil Rights, Office of the Assistant Secretary for Civil Rights, 1400 Independence Avenue, Southwest. Stop 9410. Washington, D.C. 20250. Or call toll-free at 866-632-9992 for English, or 800-877-8339 for TDD, or 866-377-8642 for English Federal Relay, or 800-845-6136 for Spanish Federal Relay. Music by Dexter Britton. Available under Creative Commons license from www.dexterbritton.com.